thank you. It's an honor to be here, and I look forward to sharing my work with all of you. I'll get right into it. I grew up reading, a lot of work by Asimov, and I was fascinated by sci-fi. And these sci-fi books talk about worlds where robots are interacting with all of us. And if you start thinking about why we don't see robots sort of around us today, you'll notice that we're still typing into machines instead of talking to them. We have machines that will create beautiful artwork and write poetry, but they won't tidy up your room and wash your dishes. We also have to scan items in barcodes instead of machines just recognizing things by seeing. And if you think about it, machines are actually failing on exactly the tasks that we're actually finding easy. So the things that we find hard, like multiplying 8-digit numbers or whatever, taking the square root of 2, those are the things where machines are doing really well. But the things that we actually do really easily are exactly the ones that that machines are unable to do. And one prime example of the fact that machines are unable to do the simplest of tasks is things like this, which all of you see on websites. Anytime you're doing an important transaction on the internet, you have to cross this barrier. And this barrier is there because to be a human being on the internet, you just need to recognize distorted letters. You don't have to believe in democracy or have good feelings for your fellow men. But machines can't do this task. And this is kind of amazing because how is it that how is it that we are not able to make machines do a task like this? And this is sort of the central goal of our lab at IISc. We try to understand how the brain solves vision. And the flip side is, of course, we'll understand how computers don't solve vision. Maybe we can make them solve vision if we understand how the brain does it. We try to do this using a number of different types of experiments. These are human experiments where we look at behavior, we look at brain imaging. We also are looking at perception in adults. More recently, we're looking at how children learn to read because new brain regions are formed when you learn to read. To understand this at the more cellular and finer grained levels, we are performing experiments with monkeys, and this is because their brains and vision is very similar to ours. We're looking there at behavioral neural activity. We are looking at how animals perceive visual information. We're looking at how these animals make decisions and so on. And a core theme that unites all of these things is computation. That is, we are trying to understand our data by building models of the data and then comparing computational models or computer vision models with the brain. So how does the brain solve vision? Well, it turns out even the brain finds vision to be a hard problem because if you look at the amount of real estate occupied by vision in the brain, almost 40% of your brain is involved in vision of one kind or the other. And that's a huge amount of brain space for a very tiny part of your body. And that's because vision is a hard problem. It's an interesting problem, and the brain also finds it non-trivial to solve it. And part of the reason we're not aware of the difficulty of the problem is because the brains have evolved to solve the problem of vision over the course of evolution, and we're just not aware of all the processing that goes on of our visual information. So, interestingly, when there is damage to various parts of the brain that are processing visual information, you can have very specific disorders where the eyes are okay, but perception is not okay. So one example of it, which we are very excited to start, is what we're calling the Indian Face Perception Project. And it turns out there's a condition called, of course, all of you know about color blindness. There is a condition called face blindness, where people with this condition have a lot of difficulty recognizing faces. And this is because of some kind of abnormal processing they have of either faces or objects or in general visual information. We don't know the reason. There is also another group of people who are super recognizers. They are actually really good, much better than normal in recognizing faces. And again, we don't understand how their face processing is better than all of us. And our goal is to try and understand this full range of face abilities. And I'm not going to talk about this project. We've just started this project. Please Google if you're interested to find what we're doing on this front. We're also looking at reading in children. And so this is, of course, an extreme condition where people are unable to read. Children have this condition called dyslexia sometimes. And this is a problem of both sound and language processing as well as visual processing. And the visual processing component of it is very poorly understood. And so we are doing some research, and more recently we're partnering with schools and nonprofits to try and understand what predicts when children learn to read fluently versus what predicts when children cannot read fluently and have lots of reading difficulties. But this is all about just a general overview. What I want to tell you about is two components in this talk. It's a bit ambitious. I'm not sure I'll go through both of them. But one component is using monkey brain recordings to understand how vision works in the real world. And the second is using human brain imaging and behavior to understand sort of novel computations that our brain is doing. So hopefully I'll go through both. So let's start with the first one. The traditional approach to understanding brain activity in animals, and especially smart animals like monkeys, is to actually bring them into the lab. And so if you want to get brain activity from an animal, you've got to kind of restrain it, you've got to bring it into a lab, make it do some task. And if you're if you're interested in studying vision, you've got to actually present stuff on the screen and they got to look at it, you need to monitor their gaze, and it's a pretty complicated setup. And actually there's a lot of different components that happens. But one thing you might be struck by, if you have never seen something like this before, is that while this may be a very interesting and sort of useful way to get information about visual processing in the brain, it's actually blinding us in a sense, because you don't know how the brain behaves in the natural world. And if you look at the 99% of vision studies which are done both on humans and animals, they're all stuff where you present stuff in front of a screen. And so one of the things that we started to do a couple of years ago is to say, well, can we actually flip this entire approach? And actually, can we study natural vision? And the vision there is to bring the lab to where the animal is. That is, in other words, bring your neuroscience to where the ecologically valid and natural conditions. But this is much, much easier said than done, because there's a lot of barriers to cross, and you can get to very uncontrolled settings where you may not be able to understand and interpret brain activity because it's natural behaviors. So we had to shed a lot of things. So for example, we need to understand and track where animals are looking at in an unrestrained settings. You got to train animals in a natural setting. You got to also do wireless recordings. And this is really crucial, because if you think about it, if you're getting information out of the brain, some wire has to go out. And unlike rats, which may not do this, monkeys will just yank the wire. And so you need to have a way of wirelessly, recording information. And this is, again, easier said than done. There's lots of power issues, electronic issues, and all that. So this is the dream team, sort of like the Ocean's 11. We're trying to understand whether we can understand brain activity in natural vision. So one question to start out with, this is sort of our monkey lab. One question that we started with saying, is it enough to just record brain activity in a natural setting, or do you need something else? Well, what we realized is that you need to have a control setting where you can record brain activity over many, many trials, because natural behavior is highly variable. And so you may not be able to interpret what you're seeing in natural behavior unless you are able to record from the brain in a more controlled setting. So we built a lab like this, which is kind of a hybrid of two settings, where the animals actually live here. It's kind of like a 5-star, hotel in a very naturalistic kind of setting. They commute through a series of doors where we don't touch them. We just kind of open doors to let them in. And then they come to a workstation very similar to ours, where we interact with stuff on a screen, and then we get some juice rewards or money rewards. In the case of our monkeys, they get a juice reward. And so basically they come here, perform some tasks, and get a juice reward. There's a pretty complicated setup behind this, because you got to track where they're looking, and you got to be able to train them on complex tasks. There's a lot of interesting anecdotes and things I can tell you offline, but basically this is the entire system. And so then you might be wondering, what do you train these animals on? So what we decided to train these animals on is a kind of fundamental cognitive task, which I think is very interesting, because it gives you sort of windows into how the brain works at different points in the task. So here's an example of the task. Basically, the task starts with the green button. The animal has to touch the green button to initiate the task. And then a sample appears and disappears after some time. And then two choices appear. So in one trial, the same object could repeat. In another trial, some other object could be shown. And the animal's task is to indicate whether the two objects that are shown are the same or different. Okay? So if they're the same, he has to press the top button. And if those two objects are different, he has to press the bottom button. So why is this an interesting task? Well, if you think about the brain activity that's evoked when you look at the object for the first time, that's just a probe for the basic visual response. It's a sensory response that's coming in. The animal just needs to hold this item in memory. Now, if you look at the delay period, the item to be remembered is gone. So there is some brain activity that the animal has to create to be able to remember that item in memory. And then the third phase is kind of a probe for decision making, because the animal has to now match what he's looking at on the screen with the content of what was in his memory. And so this is a very basic kind of cognitive task where we can probe a lot of interesting aspects of cognition. Okay, so let me show you a video. This is a video of one of our animals that performed the task a couple of years ago. So he's looking around because it's a free setup. So the animal has to do the task for getting juice. And eventually he'll say, okay, I'm going to look at the screen. And the key innovation that we had here, this is completely custom built around the animal. So the key innovation that we have here is that there's a chin rest. And so the animal has to put his head in exactly one particular position, which standardizes his eyes. And therefore we can track the gaze of the animal. And so in tasks like these, you can see the animal is actually doing the task that I told you. He's looking at two items. When the two items are the same, then he presses one button. If the two items are different, he presses another button. And then when he makes a correct response, he gets juice reward. And if he does a wrong response or moves away, he doesn't get anything. Okay, so there, for example, he's taking a break. He can do whatever he wants, but he won't get juice. And so ultimately, he wants juice, so he'll do the task. And they like this juice, so they'll do the task. Okay, so that's the setup. Okay, and where do we record from? And this is again a question that we wanted to do. What we're doing is to record from two key regions. One is a higher level visual cortex. This is the final stage of the visual processing pathway in the brain. This region is very important because if this region is damaged because of stroke or any kind of condition, you will have severe difficulties in recognizing objects. So we know that this region is very important for object recognition. The other region that we are also recording from is the premotor cortex, where we think decisions about any task are formed. And so this decision is being made in the motor cortex. So we want to understand how the visual information in the visual cortex goes off and becomes a decision in the motor cortex. Okay, so that's the overall setup. So let's run through a couple of questions. I don't have time to go through a lot of them, but the basic enterprise is like this. First of all, will this even work in a simple task? Okay, so what's the most simple task we can imagine? And we'll run that. And the second is, will it help us understand sleep? And this kind of an interesting connection between what Nitin just talked about. So let's start with the simplest task possible. What's the simplest task that you can think of in a natural environment? Well, the task that we thought of is that simply a human experimenter will walk into the room where the animal is and hand over a piece of fruit to the animal and then come back. Okay, so what do we think might be happening during this task? Well, first of all, a bunch of things happening. The animal has to look at the experimenter. He looks at the food that's being offered. The animal takes the food and all that. So that's a basic proof of concept for us. So this is many trials of the task I'm showing you here. There are two different experimenters and the three different types of food. The three foods are raisin, carrot, and banana. So we call this the RCB experiment. Okay, and this was also a successful experiment actually in this case. Okay, so what's going on here is that we also made an experiment where the animals looked at images on the screen. Okay, so we have brain activity recorded while animals are looking at images on the screen. And what we do is to build linear classifiers which look at the pattern of activity which is evoked by each particular object. So for example, I'm showing you the raisin, carrot, and banana. These are the boxes that were actually shown to the animal in the experiment. But we're taking images of those boxes and showing those objects on the screen and building a pattern classifier which will tell us if you look at when you look at the brain activity in the real world, we can then classify whether the animal is looking at the experimenter or the food and so on. So the idea is you train on the screen and then you test it in the real world. And if it works, then we've established a kind of systematic bridge between the natural setting and a kind of more controlled setting. But first of all, you've got to do a sanity check. So sanity check is, well, let's take new trials in the screen-based task and just check whether we can decode the identity of an experimenter or the identity of food that is shown on the screen. So that's exactly what we did. So the animals are just looking at these food images and experimenter images. Those are images of a few of the students who are doing the task. Then this is just the decoding type. So basically what we're doing is using neural activity to guess the identity of the food being offered, or the food being shown on the screen, or using neural activity to guess the identity of the experimenter that is actually shown on the screen. So this is all screen based. So obviously we are able to do this. This is just a sanity check. The ultimate test is of course what's happening in the natural task. And it turns out that we're able to decode food and experimenter identity correctly above chance. So we think that there's a systematic link that we're able to get between neural activity on a screen to neural activity in the real world. So will it work in a simple task? The answer is yes. This may be kind of, maybe you're wondering, well, this is pretty obvious, but what we are showing is the degree to which there's a generalization. So yes, it stands to reason that things that you show on a screen should generalize to the natural world. But in the natural task, there's lots more other influences on the brain activity. And despite the fact that all of those other things are happening, we still have a systematic relationship between the screen-based activity and the real world. So let me show you a couple of things about sleep. The big advantage of our setup is that now we can record from brain activity under any condition. So for example, the animal is taking a break. I can record brain activity and say, well, can I predict that he'll take a break? There's a naturalistic environment, so I can also record from the animals when they're sleeping. So can we understand sleep? And this is probably one of the very few studies that have been done with primates sleeping. Here's a screenshot. This is actually an infrared image of our animals, the animal in the sleep condition and the wake condition. Very interestingly, monkeys don't actually lie down to sleep, unlike us. So they actually crouch and they sleep like that. And so by looking at their body posture and the fact that their eyes are open or closed, we can tell that they're sleeping or not. We don't have rapid eye movements that we can monitor because the animals are in a natural setting. So we don't have eye movement tracking like that. Okay, so here's a snapshot of brain activity that we've recorded now from both animals. What you can see, the orange patches are the times when the animals woke up. So just like us, they kind of wake up in the middle of the night, they move around a bit, or they move a little bit during the sleep and so on. And so we annotated all of these based on the videos. And the brain activity is just given you as a sample. So the first question we wanted to ask is, well, given the brain activity in the visual cortex or the motor cortex, can I actually tell if the animal is sleeping or not? So what do we do? Again, it's basically a pattern classification approach. So what we're doing is to train classifiers on the brain activity, which is recorded during the wake state and the sleep state. And then we're saying, given a new epoch of activity, can I predict if the animal is sleeping or awake? And turns out that we can do this better than chance. But what's more interesting is that we can also predict the onset of sleep or wake states. So in other words, if you now plot the probability of the classifier or the accuracy of or the probability of decoding of the classifier around the time of when the animal is going to wake up or when the animal is going to go to sleep, what you can see is that these classifiers seem to predict when the animal is going to wake up and when the animal is going to go to sleep. And that's kind of making sense because you can imagine if you were going to sleep, your motor movements would slow down, your vision would change a bit because you're kind of closing your eyes. So there is sensory and motor consequences of entering a sleep state and also exiting a sleep state. So that all this makes sense. But what was more interesting for us, and what you're probably thinking of right now, is can we understand something about dreams? Okay, and turns out we can, kind of. So here's how it goes. So what we did was, clearly the pattern of neural activity during the wake state and the sleep state is quite different. So because I can decode the I can decode the pattern that is occurring in the brain. So then what we did was, for every epoch of neural activity, for every epoch of the whole sleep session, we can characterize a similarity between the wake state and the sleep state. And so what we're plotting here is the similarity between the brain activity recorded at every moment in time to a template wake state and a template sleep state. And so there are periods that we've highlighted in orange, which are of course the wake states, and the blue are the sleep states. But what we noticed was there was a couple of times during the sleep state where the neural activity was very much like the wake state. And that became an indication for us that maybe this is kind of like a REM sleep or dream phase, because this is known from the literature in humans and animals that there is a kind of REM phase that people go through. So we thought, okay, this is interesting. This is a REM phase that we can identify during the sleep recording. And so what they did was, the same animals were shown these pictures of these food and boxes with food and without food. So we can train a classifier that tells us whether the pattern of activity in the animal's brain is similar to when it's looking at food versus when it's not looking at food. And so what we did was, we know the brain activity in the awake state when they're looking at images on the screen, and then we are trying to decode what's happening in the sleep state based on these decoders. Okay, so that's the idea. So the hypothesis was, well, monkeys should be dreaming about food, right? And so then we said, okay, if monkeys are really dreaming about food, then the activity corresponding to when they're actually looking at the food should correspond to when they're actually dreaming about food. And so we look at the probability of the classifier to actually indicate the presence of food. So what I'm plotting on the y-axis is the probability of food as deduced from the wake state responses, right? And so what I'm simply saying here, during the NREM phase, that is normal sleep, versus the REM state, versus the awake state, what is the probability that the classifier says that the animal is thinking about food? And it turns out that in the REM state, as you kind of might expect, there's a higher probability that the pattern of activity in the brain resembles the activity that you're looking at food. You could do the same experiment on faces. You could say faces versus other objects, or objects versus other things. And those classifiers don't show you the same pattern. It suggests that whatever activity is being instantiated during the REM state is probably specific to food. And that kind of makes sense. Okay, so that's this part. And I think I will try to rush through the second part as well. So this is all about understanding real world vision. I've shown you some snippets of what we're able to find. Let me show you quickly a kind of concept of what we can do with human studies as well. So if you think about doing visual tasks, right? Here's a nice photo of our department during one of our events. And if you're looking for my face, then you're probably looking for specific features. I mean, you can look for the absence of hair, and that'll shortlist, that'll give you a short list of people to look at. But usually when you're looking for a particular, in a visual task, you're usually looking for specific features. But it turns out that we also do a lot of visual tasks which involve not looking for features. Okay, so for example, there are many tasks where we are looking for properties. So let me give you an example. I can show you these two images, and you can easily tell that the two items that are shown in the top row are actually same, and you can easily tell that these two items are different, right? You can also do a task where you're looking for the odd one out, and you can see that in the bottom array, there's an odd one out. On the top one, there's no odd one out, right? You can also do this task of detecting if an object is symmetric or not, right? So if you think about how you're doing these tasks, there is no feature I'm actually specifying for you. So how is it that you're actually doing the task? What feature are you even using? Because all of these tasks do not involve looking for any feature, but rather looking for a property in the image. So the question is, how do you look for properties in images, right? And if you're thinking that this is some sort of obscure lab-based task, I would argue that we do this kind of task all the time, more often than you would imagine. So for example, if you look at this image, you immediately know that it's Barack Obama. And you see a particular face in multiple different conditions, wearing different kinds of clothes and all that. And the fact that you're able to tell that it's the same person and not a different person is that you're actually solving a different task. Okay. You could also be looking at two animals from the same animal from different viewpoints. And again, you got to extract the features that actually help you recognize it's the same animal. These are, of course, not just about, spotting differences, and, there's all kinds of folk psychology built around all of these things. We also value symmetry. So again, when we look at beautiful temples like these in Karnataka, the aesthetic experience of symmetry is what gives you that aesthetic appeal to these things. So these are all not sort of lab tasks, and they have some relevance to the real world. So I'll just leave you the question of how do we actually solve this task. So for example, if you're looking for my face, then you're looking for some kind of similarity, and you can draw some boundary. And this is a standard approach of machine classification. There's a pattern or a feature to look for, and so you can draw a boundary, and then you found the feature. But then if it's a generic task or a property-based task, well, what's the feature space and what's the decision variable? What are you actually using to detect the features? So let me give you just a brief glimpse because I'm almost out of time. So this is the idea. The question is, how is the brain representing multi-item arrays? Turns out that we know a little bit about it. If you imagine that there are two cells in your brain, not to insult you, of course, there are many more cells in your brain. But if you have two cells in your visual cortex and they're responding differently to different images, you can imagine the response to those three images are something like that. Now we know that in the visual cortex, if you show multiple items, the response is an average of the item responses. So if you're looking at the response to the 4 monkey images, then the response is going to be just the average of those. And similarly for the horse, similarly for the dog. I'll just take a minute to sort of wrap up the idea. So what we realized is that if you're looking at pure arrays which have no odd one out, the response is going to be exactly the same as the single item responses, because you're just doing an average. Your brain is doing an average of the response. But if you're looking at items which have one odd one out, what's going on? Well, it turns out, if you think about it's true even in a vector space, if you're taking the average of the neural activity of 3 horses and 1 monkey, then the response is going to be on the line joining the horse and the monkey. Okay? And the way it will determine how much the response is closer to the horse or closer to the monkey. And so if you think about it, all these odd one out arrays are actually kind of in the middle, all those red triangles. And if you're measuring the distance of each of those arrays from the center, you'll notice that all the arrays that are actually containing odd one out are actually kind of closer to the center, and all the arrays that don't contain the odd one out are actually far away from the center. And this is the essential principle of homogeneity, which is that it's just like mixing paints. If you mix red paint with slightly off red paint, you'll still get red. But if you mix a bunch of paints, you'll start getting everything brown, right? And so that's exactly what is happening with these things. And we think the brain is computing some sort of distance from the center to solve the task. And I'll sort of stop there because I'm out of time. And thank you for your attention. And I'm happy to take questions.
Thank you so much for a very interesting talk. So I have 2 questions. The first one is, what do you exactly mean by brain activity? It must be an n-dimensional feature. So what are those features? Yeah. And secondly, what is the decoder architecture you're talking about?
So the brain activity in our case is we're recording voltage fluctuations from the extracellular medium by inserting electrodes into the brain. And so the number of electrodes determines the dimensionality of the brain activity. So if, for example, we're recording from 128 electrodes, which is the case in our case. So we have 128 numbers that you can extract at any given moment in time, which reflect the amount of brain activity which is going on at that moment in time. So for example, if you're counting action potentials, then this number of action potentials in a small time window. And so that's the dimensionality. And the classification that we use is basically linear classifiers. So we're just using linear discriminant analysis. And it's been shown, interestingly, that even if you use more sophisticated classifiers like machine learning or SVMs and so on, the classification accuracy doesn't increase too much. In fact, it doesn't increase at all. And so that's given rise to this idea that the brain is trying to encode information such that the information is linearly separable, so that you don't have to spend a lot of effort in classification, rather than you would rather invest all that effort in feature processing. That seems to be what the brain is doing.
Thank you so much.
Sir, you place the electrodes in the
Yes.
probably in the inferior temporal
Yes.
gyri.
Yes.
But in human being, I think the visual cortex is in the occipital lobe.
That's right. That's right. So that's one very important and interesting difference between the monkey visual cortex, where the visual processing in the monkey goes all the way down to the pole of the temporal lobe, whereas in the human, it only goes up to the occipital lobe, and the temporal lobe is actually involved in language and object naming and all that. There is still a visual response, but it's still mixed with language. Yeah. So that's part of the reason why in humans we don't really understand the functions of the temporal lobe very well, because those are kind of association areas. They're intermediate between vision and language. All the reading areas that are formed are in the temporal lobe and so on.
Yeah, so is there a question there? Okay. So I was just curious about where do you get like the monkeys from? Like, do you farm them or do you like pick them from?
We have a primate facility here since many years. And the animals are actually housed, and also it's a breeding colony, so we breed them.
I see. So in that case, just to follow up, like, do you think the decision making for the individuals which are like brought up in the setup compared to the ones in the natural environment would be different?
It's a great question. It's very difficult to answer also,
because I think
them in some way, and testing them will require some training.
Right. Because in the natural environment, I think there would be more factors which would lead to their decision making, right?
That's right. In fact, not just that, even if you take them from this very controlled setup, like in front of a screen, to a naturalistic setting, even that opens up all kinds of extra brain activity that's happening. It makes it very difficult to interpret even there. So yeah, I do agree with you. I think one must go in some, you've got to push systematically along the way.
Thank you so much.
I'd like to thank the Academy for hosting this symposium, and thank you all for being here.